Hello, my name is Kyle Jones and I will be discussing my project entitled Conservation of the NHX1 gene in agriculturally significant plant species and its potential role in enhancing salt tolerance. I'd first like to start by explaining the problem I am trying to solve. There is a decline in the amount of arable land. This is land that is capable of being farmed. Globally, over 7% of landmass is affected by salinity. 33% of the land that is currently farmed is impacted. The World Resources Institute estimates that we will need to produce 56% more food to support the projected global population in 25 years. Effectively, we will need to produce more food on less and worse land. I have mentioned salinity. This is the amount of free ions present in the soil or irrigation water. In agriculture, this is measured by electrical conductivity. The USDA defines a saline soil as having free ions exceeding 4 decisiemen. This is the equivalent of about 40 millimolar sodium chloride. A saline soil is a less productive soil, reducing yield and stunting plant growth. This reduced plant vigor is the result of salt stresses. There is osmotic stress, where the plant roots have difficulty pulling water from soil. This progresses to ionic stress, where ions have entered plant tissues and disrupt cellular function. This can progress all the way to oxidative stress, where these disruptions cause overproduction of reactive oxygen species, damaging cell structures, and can go as far as cell death. Of course, if this is where the story ended, there wouldn't be widespread plants or agriculture today. Plants have evolved strategies to limit the damage that salts can do. They can exclude ions, preventing them from entering the roots. They can sequester ions, packing them up where they can't do damage. They can even secrete the accumulated ions as salts through glands or pores. When studying plants, this plant, Arabidopsis thaliana, is the model species. It was the first plant to be sequenced and has been used to study fertilizers, pesticides, growth conditions, and gene functions. It is also considered to be salt sensitive, showing stress symptoms at 100 millimolar sodium chloride. Arabidopsis thaliana has a gene, NHX1, that is a sodium-hydrogen antiporter. It exchanges sodium ions in the cytoplasm for hydrogen ions in its vacuole. The hydrogen ions are much more easily managed by the cell and in this way maintains its osmotic gradient and mitigates damage from the sodium ions. Studies have been conducted to increase the transcription rate of this gene. In this particular study by Apps et al., overexpression of the NHX1 gene allowed Arabidopsis thaliana to grow in 200 millimolar sodium chloride, a concentration that heavily stunted the wild type. This is what led me to my hypothesis for this project. The NHX1 gene is conserved and functionally homologous across agriculturally significant plant species, and it represents a promising target for improving salt tolerance through genetic approaches. On to methods. I first acquired the Arabidopsis thaliana NHX1 protein sequence from NCBI. The sequence is shown here. The Protein Basic Local Alignment Search tool, pBLAST, was used to find significant alignments to the NHX1 sequence in the green plants taxonomic group. The returned list was quite extensive, which led to the need for significant sequence alignment refinement. I took the top 5,000 alignments and refined them to just the best alignment per species, reducing my results from 5,000 to 686. These were then filtered to just agriculturally significant species, plants grown for food, fiber, or medicine. This brought me down to a manageable 85 alignments. Here is a truncated list of the P-BLAST output. Notably, there are large percent identities, as well as 0.0 e-values. Additionally, the query coverage was quite high. The 85 excisions filtered and refined from the P-BLAST output were then used with the constraint-based multiple alignment tool, Cobalt, to identify conserved regions anchored to the Arabidopsis thaliana NHX1 protein sequence. Here we can see the Cobalt results. There are 2 regions of heavily conserved sequences present in all 85 alignments. If we zoom in on these conserved regions and look at the sequences themselves, we can begin to understand a little more about them. Using the Kite and Doolittle hydropathy index, we can determine the properties of these regions. The first scores a positive 5.3, while the second is positive 23.0. These positive scores mean that both regions are hydrophobic on average, with the purple regions specifically being hydrophobic amino acids. These fragments are probably part of a transmembrane domain, likely the phospholipid bilayer of the vacuole membrane itself. Further, one region shows similarities to ATP binding sites found in other proteins. Both these qualities, the hydrophobic nature and possible ATP region, indicate that the conserved regions are important to the gene's function. To summarize, the high sequence identity, extensive coverage, and extremely low e-values from P-BLAST, along with the conserved regions identified by Cobalt, strongly suggest that the NHX1 gene is not only conserved, but is homologous in many green plant species, including the diverse population that is agriculturally significant. Previous studies, such as the one I discussed earlier, have shown that modification of NHX1 transcription is sufficient to confer salt tolerance. These 2 facts lead me to conclude NHX1 is a prime candidate for improving food crops. The ubiquitous nature of NHX1 is evidence that agriculturally significant species already possess the molecular machinery necessary to fortify their salt tolerance. Simple changes to the gene's regulatory mechanisms through conventional breeding or gene editing could be sufficient to confer the phenotype. A transgene insertion is not required to improve salt tolerance. Using the computational resources available to me, I am left to conclude that my hypothesis, that the NHX1 gene is conserved and functionally homologous across agriculturally significant plant species and represents a promising target for improving salt tolerance through genetic approaches, is supported. Using each plant's native NHX1 gene with a derived or artificial promoter could increase salt tolerance without adding new protein sequences. This process could be used in all agriculturally significant species that have the conserved sequence. The proposed genetic adjustments, along with better and more sustainable farming practices, could play a large role in meeting the ever-increasing food demand and feed the world for generations to come. Thank you very much for your time. This is a project that I am very passionate about, and I hope that after listening for a bit, you understand why. If you would like further information, please look through my references and citations. Thank you again.